Good evening. And as the last speaker of the day, I promise I won't be long, but let me start by thanking the organizing committee for giving me the chance of presenting this work that was actually developed from, with the aid of several groups, both in Colombia and in Brazil. I'm sorry, it's getting... Okay, so from Brazil and from Colombia, and partly developed at the Laboratory of Advanced Analytical Techniques of the University of Los Andes. And I'm presenting it on behalf of Paula Galeano, who's former PhD students in my group, and Lida Garzón, former master students in the same group. So, the solanum lycopersicum, or the tomato, actually belongs to the Solanaceae family. And as you can see, many of the components of this family are very mostly widespread products and very highly consumed crops. Even if they're highly consumed and they have very beneficial properties, they are being attacked by several phytopathogens, costing normally $5 billion annually worldwide in the investment of chemicals to control normally the plagues. So one of the main plagues that actually attacks and causes the late blight of tomato, causing the loss of most of the harvest, is the phytophthora infestans, which is an oomycete that presents a two life cycle. So it's a hemibiotroph. It presents during the host infection. It presents an initial asymptomatic biotrophic phase, followed by a necrotrophic phase, where the disease is actually observed. So Phytophthora infestans is very difficult to control. Therefore, an early detection of the late blight would be really useful in avoiding the disease. So here, what we wanted to do was to use the LCMS and GCMS profiles associated with multivariate data analysis in the study of the interaction between the Phytophthora infestans and the tomato plants, focusing towards early detection of the disease, of the infection, and also in differentiating the different times of the infection. So part of these results have already been published, and another manuscript is under preparation. But while we were looking to have, to differentiate the different times of the infection, we started by the analysis, HPLC, MSMS, so coupling the LC to the mass spectrometer, and the solution of the Phytophthora infestans that we used was actually provided by the Microbiology Institute in Sao Paulo, Brazil. So, and the plants were infected by the inoculation of a drop of the suspension. So the controls were normally treated simply inoculating a drop of pure water. So at the different times of after post-infection, so this is HPI hours post-infection, 5 plants were randomly selected and processed for the analysis. So we obtained, we ran the analysis in the positive and negative ion mode, and what we obtained actually was that the ion chromatograms didn't show any differentiation between all the different treatments that we done. So by running an univariate analysis, so the cloud plot showed that actually the variation was about for 5,000 different features. So in the positive mode, and around the same 5,000 in the negative ion mode. So there was actually a difference. There was just not seeing, like, we were not being able to see it like simply by looking at the chromatograms. So what happened is that by using multivariate analysis, and especially like the PLC, we could kind of like differentiate the data into 3 different clusters. So we can see that it's easy to differentiate the initial control and a first cluster corresponding to the first 4 hours, like the first times after inoculation, which are the hours from 4 to 36 hours. And then we have a second cluster of infection, which correspond to the necrotrophic stage of like the infection. Then the VIP scores of the data show what are the important metabolites. So we have some of them which are obviously decreasing during the infection, and others which are actually increasing during the infection. So obviously we could think of these as eventually biomarkers of the infection, for example, in the case where they are kind of like their expression is changing during the stage. So the same results were obtained in the negative ion mode, and obviously this gave us the possibility to identify several compounds, like for example the triterpene saponines and the steroidal saponines. So these compounds, for example, normally react as to the response of the plant against the pathogen, like due to the presence of cereals in the membrane of the phytopathogen. So then these steroidal glycoalkaloids are another class of compounds which could easily be identified, especially the tomatidine here, the tomatidine, which is this one here, that could be used as a biomarker of the infection, because what happens is that normally during the stage, like the necrotrophic phase, what happens is that the mycete releases some kind of enzymes. They have the ability to break down this bond over here, so releasing the tomatidine from the alpha-tomatine. So this could be actually a very interesting biomarker of the infection. And then we also have some sesquiterpenoids, like in which already have been isolated in the infection with the same phytophthora infestans, but with the potato, and they also have normally react to biotic and abiotic stress. So altogether, we were able to identify over 106 metabolites, normally using, for example, the information given like from the exact mass and from the fragmentation patterns that in the MSMS experiments, and obviously with the help of different kind of like databases was this possible. So being able like to really see a difference in the different times that of the infection, we also wanted to see if there was a difference between different varieties of tomato plants. So we used resistant and susceptible varieties of tomato plants, and also we used two different strains of Phytophthora infestans. One which is not specific for tomatoes, so it was actually isolated from lulo, which is a tropical fruit, and the other one which we call specific, and it's actually isolated from the same tomato, so it's actually isolated from a solanum lycopersicum. These two strains were were given by the LAMFU, Laboratory of Los Andes, and also the two different plants that we use were one, the Roble species, the Roble variety, which is the resistant one, and the Santa Cruz, which is actually the susceptible variety of tomato plants. Here we used almost the same kind of like times, but we had to add kind of like another point, another treatment, which was the 240 hours post-infection. Again, the analyses were run both in the negative and positive ion mode. And again, exactly as it happens before, we were not able to see any difference among like the total ion chromatograms, but by running the univariate analysis, so the cloud plot, provide us with a very high number of molecular features for the negative ion electrospray, whereas a much lower number when it came to the positive ion mode. So we went on with the multivariate analysis, and again we ran the PLSDA analysis, and we could actually see a very kind of like 4 different groups, 4 different clusters, like where we have corresponding to the 4 different treatments that we had been running. So we have the resistant plant with the non-specific phytopathogen, the resistant plant with the specific one, the susceptible one with the resistant phytopathogen, and the susceptible plant with the specific phytopathogen. And again, we used the VIP plot to identify where like the discriminating molecular feature, having some of them which are actually increased during the infection, and others that, well, in this case, no, but then, so we can say actually that especially like concerning like the metabolic response, it is specific for every variety of like tomato plants. So it is actually depends more on the plant being resistant or being susceptible than actually more than on the phytopathogen. Then we had actually, we used the multivariate analysis here again, and it was possible to see that the 2 different phases of the infection, like between the biotrophic and the necrotrophic phase, were actually differentiated, being like, for example, in here we have with the resistant plant in non-specific phytopathogen, the change occurs like at 96 hours post-infection. So this means that the non-specific pathogen actually induced the metabolic change before, like earlier on, than what it does the specific phytopathogen to the resistant plants, which actually causes the change at the 120 hours. So it takes more time to the plant to respond to the specific phytopathogen. Given the chance that we had to see that was actually possible to detect a change in the metabolites expression during the early stage of the asymptomatic infection, we also wanted to see if it was going to be possible to determine and to have this same detection using or analyzing the volatile organic compounds emitted by the tomato plants when they were infected with Phytophthora infestans. So we again used 2 different tomato plants, one that was resistant, not specifically to Phytophthora infestans, but to several other phytopathogens, and one that was susceptible. So these plants were actually grown like in a phytotron, like with a photoperiod of 12 hours light, with a temperature and humidity that were actually controlled, and they were infected with the Phytophthora infestans. After these were the times that we selected for the infection, like for the hours post-infection, and then we decided, or we used an SPME or solid phase microextraction to be able to extract the volatile compounds from expressed from the plant and then analyze them by GCMS. So actually, or unfortunately, the results in vivo, like from the plant in vivo, didn't really show any difference again in the chromatograms. The plants was actually not infected at all or didn't show any sign of the disease even after like 192 hours post-infection, and whereas it should actually look like that, the leaf. So we were quite kind of like surprised. We tried the multivariate analysis, but again, here it was not possible to see any difference. So we had a total kind of like, was not possible to differentiate between healthy plants and infected plants. So we went on to try to see if like something wasn't working in the in vivo experiment. We tried the same experiment on the cut-off leaf. So we simply kind of like cut off the leaf and then did the same procedure like on the leaves. But again, really, it was difficult. We had infection, so we could see that the infection was going on. We did see some compounds, and but the final results were actually that it was not possible to differentiate between healthy and infected plants, whereas the differentiation was mainly between resistant and susceptible plants. The two different species, the two different varieties actually had very different volatile compounds, but we were not actually able to differentiate between healthy plants and infected plants. So, as conclusions to this work, we were the analysis by LCMS allowed actually to differentiate between healthy tomato plants and asymptomatically infected. So we were able to identify some potential biomarkers of the early infection. And also we were able to differentiate between resistant and susceptible varieties of tomato plants. On the other hand, the GCMS profiles did not allow for the differentiation between healthy and infected plants, but it was possible to discriminate the different VOCs profiles of the different plants. So, I would like to thank the students that took part to this work and all the other groups that were actually involved in this work, and all the groups, all the labs that actually participated, and Colciencia for financial support. And thank you all for listening.